This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 254. Recorded on October 28th, 2021. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from northern Michigan, Michele Swanson.
Hello, great to be with you.
We got some nice light. Listeners aren't seeing you, but you have some nice light there coming in through the window.
The sun is shining through the trees.
Very nice.
Yeah.
Very picturesque. From Charleston, South Carolina, Michael Schmidt.
Hello, everyone. We have rain. You have rain? Yes.
We had that yesterday. I had a lot of rain here. And returning for who knows how many times, always a welcome back from Tacoma, Washington, Mark Martin. Welcome back.
I'm glad to be here. I just got done teaching about shifty pathogens and the slipped misbase pairing model.
Ooh.
A shifty pathogen, is that one that does frame shifting?
Mm-hmm. In the OPA genes.
And dodges the innate or the acquired immune response.
Yes, it does. I am hoping to get students to make a little video of the process because the textbook diagrams don't really have the right flavor.
And this is Neisseria for those listeners who are curious.
You know, Mark, there's some viruses that do frame shifting.
Uh-huh.
To expose an otherwise inaccessible open reading frame.
You'll be glad to know my next lecture is on viruses, and that's just one lecture. But,
one lecture in an entire course on viruses.
Yeah, and I'm agitating that we need to have a whole course on it, but we'll see if that takes place.
You just need to get criticality in enrollment. If you can deliver 25 students, it'll be a thing. It'll be offered.
So, you know, Mark, I teach a virology course at Columbia, right?
Right.
And this semester I decided to live stream it.
Ooh, aren't you
It's
brave?
actually, I don't teach the Columbia course until the spring, but I decided to do a live streamed version on YouTube. And I do it twice a week at 11 a.m. Eastern. And I have a few hundred people from all over the world.
Oh, that's wonderful.
Who are so engaged. They're just loving it. And I give them
Vincent,
quizzes.
Is it graduate level or like introductory
No, this is
biology?
this is an undergraduate level course, but it does require, you know, a biology course ahead of time. So it's very interesting. And I think people are older than college who are listening in, and many of them struggle because they haven't had biology in many years, and they're finding it difficult, and they can't handle all the abbreviations.
Yeah, isn't that the truth? But I would think a lot of them are motivated by the pandemic.
Yes.
They are. Yeah. That has exposed their curiosity, actually, about viruses, and I'm very impressed. They ask good questions. So the nice thing about a live stream, and I will just do a minute or two here. So I can talk to them, but then they're there in the chat and they can ask questions and they chat amongst each other's for the whole lecture, right? And they can answer each other's questions sometimes, and it's fascinating. And I pause 3 or 4 times in the session to answer their questions and have a little chat with them. And I give them quizzes during the lecture. I give them quizzes every week online, and I give them a midterm and a final, and they're just loving it. Some of them say, boy, I feel like I'm back in college. This is great.
So are Columbia students also taking the class in parallel, or this is a separate offering?
This is separate. I'm just doing this for online,
great.
and in the spring I will teach my normal course in the classroom. But I wanted to do an experiment, and it coincided with being here in the incubator, our new recording studio in New York City. So I'm having a good time.
You are such a great role model.
Those pictures of the incubator look great, Vincent.
Yeah, it's a lovely space. Unfortunately, the wall behind me is blank white. I have to do something about that. But the space is shaping up very nicely. It's not quite done.
You just need to paint it that ugly color of green, and then you have a green screen. And
I like his shirt. I don't know what you're talking about.
No. If you asked me what colors Vincent's shirt is, I couldn't tell you because I am one of these color challenged individuals.
Yeah, I like green too, Michele. There's certain kinds. This is kind of a limey green. I really like.
I'm going with the more forest green, I guess.
Forest, yes, forest is good for where you're located. All right. Yes, Mark.
I had the situation where I would be on Zoom with people and I had a green screen that my wife set up for me because she's just awesome. And I had all these things to show them that were green. And if you think about it, then whatever I was showing them would take on the background. And it was, of course. And I didn't know that was taking place. That was the fun part.
Of course. Yep.
So they were disappearing.
Yes.
Or whatever he was substituting into the green screen. Yeah, I guess it would just blend into the background, right?
Yeah. I apologize, by the way. This is my little lab.
It's not. It looks very nice.
Yeah.
Not so little, Mark.
400
Not
square
so
feet.
little.
400 square feet. And you can see there's a phage painting a student did for me up there, of course.
Nice. Very nice.
Yeah. So
I think it looks like a well-lived-in laboratory, Mark. How many years have you been there?
Oh, my God. Think about this. I've been here for 16 years.
Great.
Do you know how long I've been at Columbia, Mark?
30.
39.
Wow. And you because you started when you were 10, is that right?
No. I started when I came to Columbia when I was 29, actually. It was my first and only faculty position. But I was just out of a postdoc, 29 years old. Yeah. And I've loved it. It's always been a good place for me. Yeah.
If I'm not mistaken, I was a PhD student when you arrived, and I definitely took your class.
So it was 19. When did I arrive? 82. I arrived.
So this year becomes 40 years.
Okay, so I was a couple years after that.
Well, he had to figure out how to teach. It took him 2 years to get going.
Oh, I took many years to learn how to teach, for no question. I mean, I had no experience before, and then they tell me, go teach medical students virology. And the first couple of years, I didn't even have slides. I just wrote on a chalkboard. But that worked.
Yeah.
And then slowly I got the hang of it. You can tell what works and what doesn't.
Yeah.
So I feel like I'm an okay teacher now, but it does take time. I agree. But here's the last thing I will say. I was at a medical school. I am at a medical school where teaching is not a encouraged activity, and they don't even care how you do it. They don't give you any feedback, right? Very different from undergrads, where the undergrads themselves will write evaluations that make your toes tingle, right?
Mark knows that better than most.
Oh, yes.
Yes. Anyway, we are here to talk about papers today, and thank you, Mark, for joining us. It's always a pleasure to have you. We enjoy your insight. I'm sorry Elio's not here. He would have loved to hear you.
And it's a follow-up on kind of some earlier things that we've talked about, which I'm really quite excited about.
Very good. All right. But first, a snippet from Michael.
Yes, and the paper is entitled, Graham's Stain Does Not Cross the Bacterial Cytoplasmic Membrane. This is a paper from 5 years ago, from 2015 or 2016, and it's by Wilhelm, Sheffield, Serafin, Wu, Spar, Ganella, Zhu, and Dye. And it's published in an ACS, or American Chemical Society, journal entitled Chemical Biology. And the authors begin their abstract with the following two sentences that justify our reason for covering this here on This Week in Microbiology. For well over a century, Hans Christian Graham's famous staining protocol has been the standard go-to diagnostic for characterizing unknown bacteria. Despite continuous and ubiquitous use, we now demonstrate that the current understanding of the molecular mechanism for this differential stain is largely incorrect. So I became aware of this paper due to my subscription to the Micro EDU listserv, which you can access if you're a member of the American Society for Microbiology. And there were two postings that came across my desk at the time I pitched this paper to my co-host, and I thought it would generate more discussion on the listserv. But when I checked back yesterday, there were still only 2 postings. But I'll put into the show notes how you, if you're interested, can subscribe to any of ASM listservs if you happen to be a member. The other reason it made my cut is Michele remarked recently that she's in the process of revising the microbe textbook. So perhaps this paper and a discussion of how we refine our thinking as to how new information can actually displace dogma warrants some time for us to go over it on TWiM. The gold standard of the gram stain the authors refer to in their narrative, or the mechanism of the behind how the gram stain works, was originally expanded upon by Julian Davies, former ASM president and a friend of Elio's. And I thought he would be here to insert a colorful Julian Davies story. So I apologize that Elio's not here to add some color to this. But you can find these 2 papers in the Journal of Bacteriology, and I also place those 2 papers in the show notes. And because ASM makes its papers available after a year, you can, of course, download and see what Dr. Davies and his team discovered. For those of us who teach this differential staining technique or actually perform it on a daily basis, like the clinical laboratorians, we can all likely conduct this protocol by heart. And it consists of a series of time-sensitive steps. But for those who don't routinely do this, I'll review those steps, as understanding the steps is important for understanding the results from this paper. And they are actually important in understanding how we are actually refining the mechanism. And the way these authors effectively displace the dogma of over 40 years is through some really cool microscopic techniques. Now, the first step of a gram stain is not actually a step, but it involves the selection of the cells that you will stain and how you will attach them to the glass slide. I was always taught to use microbes from a freshly grown plate or actively growing cells if you're using a broth-based culture. If you're using a culture that's too old or it's not in the right growth state, you won't really get a good gram stain. And the second thing is you have to use a clean, grease-free glass slide. So the protoplasm of the cell actually adheres to the glass slide when you do that heat fixing step. The other thing that I was taught by an anaerobic microbiologist, Mike Cox, is he said if you're staining strict anaerobes, in order to get a good gram stain, you got to do that gram stain in an anaerobic chamber. And that statement is really true. You do need to do anaerobic gram stains in an anaerobic chamber. But this paper actually offers us an explanation as to why it needs to be done. So regardless of aerobe or anaerobe, your best smears are not too thick, not too thin. And Mark probably has frustrations trying to teach the Goldilocks effect to students. And then the other thing is to allow that smear to naturally evaporate all of the liquid. And once it's gone, you gently heat fix the protoplasm of the microbe to the glass slide by gently passing it through a flame or putting it on one of these heated slide surfaces that will actually heat fix. The trick here is not to poach the cells. I remember my advisor said to me, "You're not making poached eggs. You don't boil the protoplasm onto the slide."
Mark, do you teach this every year to undergrads?
So I'm not a big gram stainer. I've done it. I find that when students do it, we end up with purple stains everywhere. And so what I've done the last couple of years is I've done the 2% KOH test. Are you familiar with that? So it turns out that, and again, you know, it's one of those things that people knew about a million years ago and it fell by the wayside. I believe that you'll see, and I'll look up the reference if you'd like. from the 1970s that if you put a drop of 2% KOH on a slide, you pick up a toothpick with an organism and you kind of smear it in the KOH and then you lift, the kind of string that you get means that it's gram negative. And the lack of string is gram positive.
Wow.
Oh, that's easy and fast.
Yeah.
And they're dead. You don't have to worry about contaminating anyone.
That's true.
But you don't get to look at them under the microscope and look at the architecture and configurations.
True. You can't do that. And like I've said, when I teach gram staining, I do it one-on-one with students in my lab because in a big group, I just can't imagine the mess.
Yeah. We teach it to our first year medical students in our small groups. So we'll have to change our course pack after reading this paper.
So step 2 is the addition of the primary stain, crystal violet, which is a triphenyl methane-based dye, or Alan Dove and his TWiM colleagues often refer such molecules as polycyclic chicken wire. But to anyone who has ever stained their clothes or fingers, it's nothing more than a brilliant and intense colored synthetic organic dye that, are based on these hydrocarbon structures, and this happens to be triphenyl methane. The result is that the positively charged crystal violet molecule passively diffuses into the cell, where it then electrostatically binds to any and all available anionic, anything with a negative charge, is fair game. You then rinse the primary stain from the slide with water and then apply the Mordant, which is typically a substance that's an inorganic oxide that then combines with the triphenyl methane-based dye or stain and fixes it to the material. Since we are recording this episode a few days before Halloween, it's almost like a potion from Harry Potter. But the Mordant here is Lugo's iodine, which is potassium iodine 2 grams and iodine 1 gram and 300 mils of distilled water. And it literally bonds that crystal violet iodine matrix into the peptidoglycan structure. Most of us use this Mordant as our first reagent in our middle school science projects to test for starch. Since I teach dental students, I use Lugo iodine to help them understand and visualize the mucogingival junction in the mouth. Similar to the method of staining apples or starch, you can actually stain the alveolar mucosa. It has a higher glycogen content that gives a positive iodine reaction versus the keratinized gingiva. So you can even use iodine in fancy medical application. So here the iodine reacts with the cationic crystal violet, yielding this crystal violet Mordant precipitate. The next step, and is the one that is often most challenging for novices to this differential staining program, and the trick is to watch the droplet of the decolorizing agent. And the decolorizing agent here is 95% ethanol. So as you know, ethanol is organic solvent. It's dehydrating. And what it does is it will strip the crystal violet complex out of the cells. And the old dogma specified that the crystal violet would actually go all the way into the cytoplasm. They're going to disrupt our understanding of that and actually teach us that the crystal violet barely gets through the peptidoglycan matrix. And again, in between each step is a water rinse to stop the action of the previous step. And the final step is the addition of a counterstain. Generally, it's one that's less intense in color. And in the gram stain, that happens to be safranin, another positively charged dye, but simply a different color. And that's what gives the cells its alternate pink color. You then blot the stain slide and observe a number of fields under an oil immersion lens in order to see whether your cells were gram positive, that beautiful violet color, or the pink color that was conferred to the gram negative community. So with the mechanics of the protocol explained, now it's up to explain why the protocol works and why I was taught to use fresh cells and if working with anaerobes, to stain them in an anaerobic chamber. So our current understanding of how this stain works stems again from Julian Davies and his co-workers, who employed energy dispersive X-ray spectroscopy using scanning transmission electron microscopy to actually follow the dye. Davies and colleagues offered that both crystal violet and the Mordant freely cross the three main cellular barriers in gram negatives: the outer membrane, the peptidoglycan mesh, which is the biomolecular layer in the case of gram negatives, and then the cytoplasmic membrane, resulting in the accumulation of this crystal violet precipitin within the periplasm and the cytosol. The alcohol then would denature the proteins, dissolve lipid membranes, and the decolorization step would attack the structural integrity of both the outer membrane and the cytoplasmic membrane. Of significance is in the gram negatives that as the outer membrane is covalently bound to the peptidoglycan mesh, the peptidoglycan mesh being much thinner, only bimolecular in the gram negatives, it would be disrupted and torn away. Conversely, the thick peptidoglycan layer of gram-positive species is only slightly perturbed by the alcohol solvent, resulting in local bulges and deformation, but largely remains preserved. Thus, the current understanding is that as the crystal violet precipitate stain is readily washed out of the perforated peptidoglycan mesh of the gram negatives, but it's retained by the intact peptidoglycan mesh of the gram positives. Now, this paper upsets the apple cart. It will demonstrate that the dogma we were all taught, that crystal violet crosses all cellular barriers, is not actually true, because the triphenylmethane-based dye crystal violet only can slowly diffuse across the peptidoglycan mesh and never penetrates the cytoplasmic membrane, regardless of whether or not it's gram negative or gram positive. And that's what the experiments are all about. They offer that the transport of the molecule across living cell membranes can be observed with real-time resolution, and this is the fancy term, through time-resolved second harmonic light scattering and bright field optical transmission. Don't ask me how this works, because it's Halloween, so I'll invoke magic. And it really was a very cool set of experiments. And the figure that they show in this time-resolved transmission microscopy, which I can explain, you're looking through a microscope and you're seeing color shifts. But the spectrometry that they're explaining is really pure chemistry. And I'll leave that to the explanation of the authors, who do a very nice job explaining it. So they present direct evidence proving that crystal violet is incapable of crossing the cytoplasmic membrane. And they do this with a clever control. They use another triphenylmethane-based dye, malachite green, and Mark probably teaches spore stains, which, if my memory serves, is what's used in a spore stain. And the malachite green actually has a different kinetic profile. It actually goes wicked fast through the outer membrane, the peptidoglycan, and it's much faster, while crystal violet is much, much slower. Their first result that they offer is pretty convincing. You know, they're looking at malachite green kinetics. It's structurally similar to the gram stain crystal violet dye. And in their time-resolved transmission, their microscopy, they show that the crystal violet also saturates the bacteria, but in a mere 2-minute, the malachite green is approximately 4 times faster than the crystal violet. And their harmonic light scattering data revealed only a single transport peak, the purple curve if you're looking at the paper online, and it's identical to the fast transport peak for malachite green and is suggestive that the malachite green is capable of rapid transport across the water-filled porin channels that are embedded in the gram-negative E. coli that they happen to be studying. However, following fast transport across the outer membrane, the crystal violet exhibits significantly slower penetration into the peptidoglycan mesh. This is diffusion-limited absorption. If you will, the outer leaflet of the cytoplasmic membrane is not going to let it in. It's effectively excluding it, and their harmonic light scattering is effectively able to control it. And when I first read this paper, I thought, well, they're doing this on live cells. They're not heat fixing it like was done in the traditional gram stain. Does heat fixing screw things up? Well, that's their next experiment. They show that heat fixing has no effect on the differences observed between crystal violet and the malachite green. So we throw heat fixing out the window. And as this is a chemistry paper, they had their next series of experiments are a long set of equilibrium equations that are supported by exquisite data from the second harmonic light scattering signal processing that they do that actually culminates in a kinetic model figure in which they actually show the differences in the bacterial membranes and the bacterial cell wall structure. They offer us the speed kinetic, and what you see is that the crystal violet is simply just that much slower at penetrating as opposed to the malachite green. So their kinetic model of their basic conclusion, namely that the crystal violet is unable to cross the bacterial cytoplasmic membrane, and it's principally based on the time scale of the traditional gram staining protocol that argues simply that gram-positive cells kinetically trap the crystal violet within the peptidoglycan matrix, and because gram-positives have a much thicker peptidoglycan matrix, more is actually trapped, while the biomolecular layer of the peptidoglycan of gram-negatives fails to engage since there is no matrix. Their critique of the Davies data in their discussion really requires you to go back and compare and contrast. And here is where it really requires you to have read all of those papers. But I think they still make their case because they're using complementary methods of the second harmonic light scattering and looking at it through the scope, and they are quantitatively characterizing the real-time bacterial uptake of crystal violet, that they were able to make a convincing argument, at least to me, that the dogma that crystal violet is unable to cross the bacterial membrane is indeed true. I mean, Davies was wrong in his understanding of how they did those experiments. And on the time scale of the gram stain protocol, they offer that the crystal violet is barely able to diffuse beyond the comparatively thin peptidoglycan layer of gram negatives, given the substantially thicker peptidoglycan layer of gram positives. So the crystal violet is invariably kinetically trapped within the mesh. So the dogma that I have been principally teaching, that the Mordant and the crystal violet makes an insoluble precipitant that makes it really challenging for the alcohol to wash it out of gram-positive cells, still holds. I never bothered the medical students or the dental students with an understanding that the crystal violet was going all the way inside of the cell, and that the addition of the crystal violet and the iodine results in the generation of this precipitant, which is largely isolated in this peptidoglycan matrix. But then here's where our dogma begins to change a little bit from the Davies to the current understanding. Following the destructive alcohol wash, and this is why you need to use fresh cells or actively growing cells, and why anaerobes work best in an anaerobic chamber, is that the destructive alcohol wash actually rips the peptidoglycan matrix of the gram-negative cells off and perforates the peptidoglycan matrix of a gram-positive cell, and then the alcohol allows it to be effectively washed away. That magic drop at the end of the slide that we're all taught, when it changes from that purple color to the clear color, that is when you stop the alcohol decolorization. If you add too much alcohol, what will happen is you will, again, even in the case of gram-positives, over-decolorize, which is effectively ripping the peptidoglycan off of its attachment on the cytoplasmic membrane. And the more alcohol, the more that is ripped away. Their conclusion that the Gram-Stain protocol still works is comforting, though it may cause a bit of a kerfuffle amongst those who teach gram-staining. So summing things up, the authors used really cool complementary methods of time-resolved second harmonic light scattering and transmission microscopy to quantitatively characterize the membrane-specific uptake of the gram-stain. And in contrast to previous dogma, we now know that the crystal violet Mordant complex is locked into the peptidoglycan matrix, doesn't get inside of the cell, and that the gram-stain works principally because the peptidoglycan matrix of gram-positives is simply thicker or more stable, able to withstand the alcohol decolorization step. And should the peptidoglycan matrix be destroyed, or you use old or damaged or anaerobic cells in an aerobic environment, which can lead to lysis, the protocol won't work because the cells are no longer intact. And finally, at a minimum, it's always good to acknowledge our colleagues who are introducing to the next generation our molecular understanding of defining one of the most fundamental cytological procedures of how our discipline is both explained and worked. And they did this in an elegant set of experiments. And if we are get our next generation to advance our discipline, we have to at least help them understand why stuff works before we reduce everything to a computer file. So that's basically the paper, folks.
So, Michael, does this make any difference in practice of gram-stains?
No, none whatsoever. It just is a little bit of a shift to dogma, which is probably why the undergraduate community who routinely teach gram-stains to the next generation haven't gone crazy over the shift in dogma. But back in the 80s, when I was being trained and the two papers from Davies came out, it was quite big news. And I was hoping Elio would be here to add some color to that story because it was big news in the 80s. I remember discussing them in Journal Club, which is why I was intrigued when the paper came across my email in the listserv.
But certainly the teaching of the gram-stain needs to change to accommodate this, right?
Yes. It should at least acknowledge that what's actually going on. And it's really the peptidoglycan mesh and that insoluble complex that's created, akin to the way if you go back and you look at the derivation of the word mordant, this is in medieval times of how the people who dyed fabrics, they would have this magic chemical, and it was literally potassium iodide that would fix the dye to the copper fiber or the flax or whatever they happened to be using.
And speaking of Mordant, the other usage is a Mordant sense of humor, a really biting sense of humor. So it's, grabbing on to the dye.
Yes.
I can't help thinking of Mordor when I hear that word.
Yes.
Merlin and more. I mean, it really is pretty neat.
I'll say it's not often that we read a paper where they're using really state-of-the-art quantitative technology, and yet their first two references are in 1883 and 1884 by Hans Christian Graham, who was a medical student at the time.
I put that into the show notes, his original paper.
Did you translate it for us?
No, ASM has it translated, and the name of the original paper translated into English is Differential Staining of Schizomycetes in Tissue Sections in Dried Preparations. Now, ASM on their website has the link to that paper, but it's a dead link on the ASM website. So I alerted Ray, our esteemed producer, to see if he can surface it out of the archives of the ASM's website.
So can I jump in and say
Please.
that I'm going to put the 2% KOH reference on the show notes for people who might be interested.
Yes.
Now, there's a difference between gram staining to discriminate between gram positives and gram negatives. And, of course, the fact that staining in general allows you to visualize microbes. So let's make that clear. I also visited Julian Davies a couple of years ago at UBC.
Oh, here's Elio now.
Elio. And I just wanted to say Julian Davies is one of my favorite people. And he told me this story, and Elio, I'm glad you're here for this, that he was trying to fix Thiobacillus to a glass slide. And no one had told him that when he did that, it would just concentrate sulfuric acid.
And dissolve the slide.
So you would just got a very clean slide is the way. There was nothing to look at, which was just darling. The second thing that I wanted to say is I use crystal violet all the time to measure biofilms.
Oh, yeah.
So, I mean, that's what I use Lugol's iodine to look for, amylase activity and things like that. So most of the components have good use elsewhere. May I finally say that is such a definitive and clear and exhaustive review that I feel really terrified of my own talk here in a minute.
No.
Hi, Elio.
Hi there.
Welcome.
Thank you. Sorry about that.
No worries.
Okay. Quite all right. Good to see you.
Do you have a Julian Davies color story?
Gosh. He's a very colorful character, but I can't think of one.
Okay.
We were talking about his two seminal papers from the mid-'80s where he defines how the gram stain worked. And the paper that we discussed in today's snippet sort of disrupted the dogma that has stood the test of time for the last 40 years and is actually not really changing it all that much, but changing it enough that the crystal violet doesn't penetrate into the cytoplasm, which Davies had established.
Yeah, and so it wasn't the test of time, yes, but not a direct test of the interpretation of how the stain worked.
All right, Mark, you're up.
All right. Well, by the way, I should mention, because Elio's presence reminded me that when I was talking with Julian many years ago, and he was talking about the constellation of small molecules, he got very interested in that as a monitor for things. And he said he wanted his own ohm. And so I thought of the Latin parvus, meaning small. And so I started calling that the parvome with him, which he used several times. So that was
very good. It's a pretty good term.
Yeah, I thought that. Do you mind if I just give a shout out to my Bio 350 Micronauts?
Of course.
I'm going to have them listen to this. And
Good.
I just want to say how proud I am of all of them, because on campus, my friends, 98.7 of our students are vaccinated, fully vaccinated.
Great.
96.3 of our faculty and staff, and we have 0.3% positivity.
Wow.
In the state of Washington, 72.5% of us are fully vaccinated, and I am waiting on my booster. So I think we're doing a good job here. I'm hoping.
Great.
So my students are amazing, and I just wanted to point this out during this whole mess that we've been in. When we finally came back to campus, distanced and masked, they're so anxious to work with me about things. I just love the fact that they're willing to interact with me and ask questions about things. When I talked about slipped misspaced pairing today in class, they really got right to the extensive missense versus early nonsense, and it was just great. So fingers crossed is what I'm trying to say. Today, what I'd like to talk a little bit with you about is a follow-up on something that we've talked about before. And this is from the laboratory of, and I want to make sure I pronounce this correctly, Batbeleg Bor, who describes himself as Mongolian, so I'm very fascinated. He's at the Forsyth Institute. The paper itself's title is Episymbiotic Saccharibacteria Suppresses Gingival Inflammation and Bone Loss in Mice through Host Bacterial Modulation. And I have to tell you, I was a big convert to the shifting and drifting paradigms of our esteemed colleague Elio Schaechter for many years. And the most interesting thing to me is sociomicrobiology. I come to that by looking at predatory bacteria originally, but I want to just point out for everybody else that this idea of microbes attaching to other microbes and doing things is a real growth industry, if you'll forgive the pun. Years ago, I actually did an essay in Small Things Considered on chromochromation aggregatum, where the small green epibionts are able to direct a large central rod toward phototropism, which is just fascinating. In addition, Jo Handelsman's work with what she's calling now the Thor assemblage, Bacillus cereus, thick peptidoglycan layer, and flavobacteria living within that peptidoglycan layer, and actually making things that break down peptidoglycan. I wanted to call them peptidoglycan surfers, and that's probably not, that's not cool.
Surfers. Good them.
Lighting bacteria right there in the peptidoglycan. And what I'm trying to say with all of this is the idea of microbes attaching to microbes and doing things is all over the place when you look. And my suspicion is we're going to see it everywhere. And this paper today, I think, really illustrates that point. It's not just Carl Stetter with Ignococcus, and it's oh gosh, what was the name of the little epibiont on that? Nano something or the other. But my point is, this idea is everywhere and getting more and more interesting.
So it's not just bacteriophage that are getting into bacteria and altering their phenotype.
Right. And this idea of how they communicate is of great interest to me. The paper itself is of a format that I think is very useful for students. And what I like about it is it has an in brief, it has highlights, it has a graphical abstract. And so the way that I want to just say right now, because I'm being very cognizant of time, so the real take-home lessons are something that we kind of alluded to before when we talked about these ultra-small bacteria in the past. And we talked about, in fact, work done by the Bohr Lab in the past on this. And there's a particular type that can be an epibiont that lives in the surface of dental actinobacteria. And they can find multiple isolates from this. So this idea of creating assemblages is more and more going to be a growth industry. And what it appears to do is different than what you would expect. They tend to find a lot of these ultra-small bacteria with the actinobacteria. They're, what would you say? They're chariots, for lack of a better term. Mm. You find a lot of them when you're looking at in the gingival pockets of periodontitis. I can't even say that, Michael. Would you help me?
No. Just refer to it the way the perio guys do. They just call it perio pockets.
So perio pockets. And you would expect that it has something to do with the inflammation, but that's not true. What it looks like in this paper, and this is very exciting to me, is it downregulates particular pathogenicity factors from its larger host. And we have so much to talk about with regard to that. If you look, in fact, at the graphical abstract, they even have cute pictures of mice.
Cute pictures of mice.
Yeah. We have mice, graphical mice with and without the host bacteria and the smaller member. And I just think it's a fabulous thing to look at. But, Michael, I don't mean to put you on the spot, But can you talk with me a little bit about the ligatures that they're doing to create to enhance the periodontal disease?
They're effectively restricting, and that restriction then results in the disease process accelerating. And they're doing it in the cute little mice, because mice continuously make teeth, unlike people. And I'm trying to figure out, Mark, where's that figure of it they actually show?
They have a nice cartoon of it. Thank goodness it's a cartoon.
Yeah, that's
reminded me of dental floss run amok.
Yes, that's effectively what it is. And the periodontist actually uses these ligatures sometimes that are impregnated with antimicrobials, antibiotics like tetracycline, and they slowly leach out into the space to alter the perio pocket and to try to reduce the inflammatory bacteria. But this paper is so exciting because they're using other bacteria to do that for them. I mean, this argues strongly, never use antibiotics.
A probiotic, huh?
Yeah.
Figure 3, Michael.
Yeah, I just have it in front of me. I knew I read it. It's just, it looks like they're tying a piece of dental floss around a tooth, Elio.
And what's interesting about that is that, they have baseline observations where they just put I'm going to call it the dental floss from hell.
That's fine.
When they put the dental floss from hell, and that's like basal level of damage, and then when they also add bacteria to the dental floss from hell, and then they do the bacteria with the episymbionts, and are able to actually put together some wonderful statistics. This is a very thorough paper, in my opinion, on this subject. And
what is in cell host and microbe?
Yeah, well, I like it. And I have this question for those of you who are medical microbiologists, because I'm curious. When the comment was made in this paper several times that the TM7 type of microbes, which they're starting to call Saccharibacteria, and they're everywhere, when they seem to say that they're like inflammophilic, they like to be around inflammatory situations. Does that mean just leakage from dead or dying cells that people suspect that they're using? Or are there other types of things that are being secreted under those conditions?
We know in periodontitis that smoking, which is naturally inflammatory, can create that sort of environment. And in fact, there are a large number of studies out there specifically getting at how smokers have really scary teeth. In terms of, and you can actually sort of go up to people and do this experiment by assessing for halitosis and how bad their breath is. And as the periodontal pocket depth increase, so does the malodor. And the malodor is a reflection of the short-chain fatty acids that the anaerobes in that low EH environment are fermenting, and they're all feeding each other. One man's fatty acid waste is another man's breakfast in this continuum of microbial complexity.
Or if somebody once said, better halitosis than no breath at all.
That's true. That is true.
But to get back to your question, Mark, so it's not just that you've got lots of neutrophils in there. It is a change in the profile of short-chain fatty acids that other bugs can use then as nutrients.
Because in the paper, they talk about finding these episymbionts, and I don't know if I like the term basymbionts with all respect to the authors. 100% respect to the authors on this. But I'm reminded a little bit about getting lost in the nomenclature by this, but it's fine. So we have, if you want to call it a chariot like I was versus a rider, we can do that. But the point that I'm getting at is in a number of pro-inflammatory situations, they find these microbes. And they're finding them all over the place when you start looking into records of microbiota in different sorts of environments. It was really surprising to me. It's easy for me to look at the mouth ecosystem and think of it like, the rainforest, as people often do with the gut, certainly compared to the skin. But it seems to me we're going to be finding these wherever we look. And when I'm teaching my students, I talk about the microbial perspective being difficult, and I talk about the 3 deadly centrisms. The first is eukaryocentrism, the second is oxycentrism, and the third is colicentrism. And darn it all, what I really like about this paper is that I've done techniques like a lot of these with predatory bacteria, and it's working for these. For example, how do you isolate these small writers, as it were? You passage them through a 0.45 micron filter, syringe filter, like I have in my lab. And that is the way that I separate Delavibrio from prey cells. And this is exactly what they do when they created genome sequences. Although, as I think we've talked about before, small genomes for these writers, as it were, I want to call it maybe 700, 800 kb, maybe 800 genes, it looks like. And they're missing all kinds of things. So my thought is that they really are using materials that their chariot is providing, these actinobacteria. So I'm reminded of something Arturo Casadevall says, is that he doesn't like the word pathogen, right? And one of my students made a joke and called it pathogon, and he liked that. But it all comes down to, as real estate agents say, location, location, location. I would come back to context, context, context. And the reason that this is important here is that they make the comment in the paper several times that they would have expected a lot of the TM7 type of ultra-small bacteria to be enhancing inflammatory responses, and they were kind of surprised that they didn't. And again, the microbial perspective might be very different than our interpretations. And so there might be kind of a Goldilocks area where they certainly are inflammophilic, but they don't want to make too much or enhance too much of an inflammatory response. And in that regard, turning it down is fascinating.
Because what you don't want is pus. Pus brings white blood cells by the billions, and they come in and they will eat and consume all of the community that are hanging around. And so you truly do want the Goldilocks effect. And I think in our gut, as well as in our mouths, this dynamic equilibrium of it is akin to a predator-prey relationship or a parasite-host relationship, where they're literally the camp followers. You have the ones that are effectively controlling, but then there are these other players that are responsible for the depth of the community that is actually present and all of the wonder. And the consequence in some cases is periodontal disease or alternatively good health.
It's so funny because I'm looking here on page 3, and there's a wonderful bit when they actually probe some of the perio pockets. And thank you for that again. And they're finding between 40 and 20% of what they find there on a molecular basis are the TM7 types. And that's very interesting to me because, your automatic knee-jerk reaction or knee flagellar reaction, whatever you want to call it, was that they're enhancing the inflammatory response. But it doesn't look like that. And that, to me, is the most fascinating thing. I also sent to Vincent, a nice picture I got from Professor Bohr's website of a nice SEM showing the association, which always fascinates me. Certainly, Figure 2 has some really nice pictures using...
well, it's phase contrast, and it looks like...
fish. They're using fish. To be able to show TM7 types and the actinobacteria. And then they have some nice phase contrast microscopy as well, which is good. One of the things that I wanted to draw your attention to is that I love this idea. They started to bait to find them. So they would actually take a potential host and mix it with some of these contents, and then they would start looking for them. Now, what's interesting to me is some cases, not all, they would get a negative effect on the hosts and have a population crash. And then they would rebound from that. And to me, this sounds like the evolution that we expect to take place. And I hate saying toward a particular state, but you know what I mean, towards survivorship in any event. And that's exactly what we're tending to find here after the baiting. I look at this work and I'm thinking, I cannot imagine going through the process of doing the baiting and then just waiting to see what's there. And yet when they did it, they were able to commonly pull these things out of different environments, just exactly this way. And again, to separate them back again is to pass them through a filter. To me, I'm wondering whether the associations are not permanent, but more peripheral, so that they are able to come, let go and come back and let go and come back. I would love to see connections, but I believe that's going to require some of the really fancy work that are done with the machines that cost millions of dollars, right? For the really fancy cryoelectric tomography techniques. So what they were able to show that really amazed me is they did pairwise like associations with these ligatures. And again, as I mentioned at the beginning, they were able to show that no matter how they made the ligatures, and they would have a baseline level of bone destruction or erosion, for lack of a better term. But if they pre-treated the ligatures with one of several actinobacterial strains, they would get very much enhanced difficulty. But if they also used the association, it was attenuated. You had less of that taking place. To me, that was really fascinating. And it looked to me like it was a general thing.
Well, bone loss is generally strongly stimulated by the immune system. The immune system can cause bone reabsorption. And what these passenger microbes, for lack of a better word, what they're doing is they're saying, we like it here. We don't want you to lose the bone and drop the tooth. And in mice, because mice continuously generate new teeth, unlike we only get 2 shots at the deal. And it's really pretty fascinating that this ligature, and what the ligature really does is just keeps debris up there, right, so that you're continuously feeding the little creatures. Just don't floss your teeth for a few days and then floss and see what you get out. Or better yet, just smell your fingers around the floss and you'll get the short chain fatty acid. Well, most people don't appreciate what's actually going on. And once you understand the waste products are short chain fatty acids, and you have a very sophisticated gas chromatogram in your nose that can discriminate, and you'll be surprised at your ability to detect good oral health and bad oral health. Because everybody knows what bad breath smells like.
Yeah. So let's
and that's principally from Fusobacterium.
Let's get back to this paper. I agree with you. It was very comprehensive. They even went in and did some, looked at the outer membrane protein profile and then identified a couple of key players, didn't they?
Collagen binding is one of them.
Yes.
And the ability to use sialic acid. And this is something they were used to. And it kind of fit their model. And I guess I'm impressed because no matter how many times we say we don't have a bias when we look at research, you would not want to think that this thing that you find in inflamed tissue would be moderating instead of enhancing the negative effect. But it looks like that's the case, which to me is fascinating. In addition, Michele, I don't know if you saw this on figure 5. Of course you did, because you see everything. I loved it when they were able to find colonies of the actinobacteria, the irregular ones, that are carrying these things on their surface as hitchhikers. Yeah. And they could identify them by colony morphology.
That's gold as a bacteriologist. When you see colony morphology, it makes it gives you a real handle on the biology.
I mean, when I'm looking at, I guess, part C of figure 5, and they have this beautiful, wonderful PCR versus what the colony they're seeing. This is exactly like Joe Handelsman's model with Bacillus cereus and the flavobacteria, by the way. and it is because the ultra small bacteria cannot grow by themselves, which I'm used to from the things that I look at, right? So that was wonderful. But again, they brought to bear so many instruments on this, and I was so impressed by every level of it. Even to the point of trying to make deletions in some of the genes that are involved in the actinobacteria to see what effect they would have to mimic what the partners were doing. What do you think?
I think this is a brave new world that is going to change dogma in the perio community. And what was once considered a frank pathogen now is going to be considered, if you will, a commensal.
Yeah.
Because of its ability to dynamically interact with the inflammatory process and control the inflammatory process.
Yeah, even a beneficial microbe even.
Yeah. Yeah, it really becomes, it really sets up dogma. There's an old dental truism in terms of it was called the Kinetic Model of periodontal disease, in which they use colors and a concept of succession. And they start out with the yellow complex, which is principally streptococci, which are responsible for the initial infection for dental caries. And then they go all the way up to the red microbes, which are Tannerella, Fusobacterium, Treponema, and Porphyromonas gingivalis. Fusobacterium is not in the red zone. And it again illustrates that what we were initially thinking now is even more complicated than we ever thought.
I would agree. I don't know. When you use the word, Michael, when you use the word commensal, are you using it in the biological sense, or is there a dental sense that you're using?
I'm using it in the biological sense, where it's beneficial to the host and as well as the microbe.
Yeah, so mutualism is the word that I tend to think of a little bit in that regard, because I believe that they're both getting something out of the association. And I think the part that I hate saying blew me away, but it did, is that you can bait for these. So in other words, these TM7 type organisms, these ultra small bacteria, seem preset to form associations with other bacteria. And they are able to independently cultivate just a couple of types. They've got one here, and then there was the TM7X that was done from human saliva a while back. But my guess is the more we look, the more we're going to see. And it reminds me a little bit of bacteriophages when I was in college. And we thought, well, they're just kind of an interesting lab effect. And then when I was in grad school, they might have some kind of effect in certain environments. And now they're very important in the ecosystem.
But it's been tricky to study because it's been hard to culture them independently.
Exactly
right?
so. Exactly so. And like even though I abhor coli centricity, I would argue that when we have a good model system like that, we can begin to expand from it and say, can we find other types of associations like this? Keep in mind that Delovibrio has quite a large genome. It does not have this like Darwinoid genome that you expect from some types of mutualists of this nature. So this is much closer to what I would expect between an association of large and small together. And because of the ubiquity that you see in other inflammatory environments in the human body, I'm guessing we're going to see it everywhere. And that to me makes this extremely fascinating. Not just something for the teeth, is what I'm saying.
And an opportunity to for intervention and promote health in a new way.
And again, coming back to one of the experiments when they had the two target genes that they thought were being downregulated by the TM7, when they were able to manipulate the larger partner to obliterate those genes through knockdowns or knockouts and show that they also showed reduction in inflammation, that's really gold to me.
So what's in it for the pathogen? Why doesn't it overcome this?
Yeah.
In some way. I mean, is it inhibiting its dissemination in any way? Its division? Are the levels lower in the presence of TM7?
So they made comment in some of the associations they would find hundreds and hundreds of episymbionts, but they didn't say that for all of them. And my guess, and it's only a guess, is that after multiple passages, you end to find out where they've kind of streamlined their association. And I'm really worried about that because evolution does not have a direction.
Sure.
And it's so easy to fall into that. I would prefer to think that there's like a preset in this particular clade, taxon, whatever you want to call it. It's preset to have associations with other microbes. Hitchhikers.
Sure. Yeah, I mean, it has to be neutral, right? Because otherwise one is eliminated eventually.
Natural selection is a very jealous mistress.
Now, the reduction of inflammation, that doesn't compromise the host in any way. I mean, I realize that pathology is often inflammatory driven, but on the other hand, it can clear pathogens. So it's not interfering with the clearance, right?
It's like a delicately sipping parasite. You know what I mean? It's not taking too much.
I got it.
It's like the bats that have very sharp teeth that slit the vein of the animal and then lick up the blood. Well, it's Halloween. They lick up the blood from the animal, the vampire bats, that they just have very sharp teeth. They slit the vein and then the blood just leaches out and they just lap it up like a dog licking water out of a bowl and until it clots.
Well, much of the late disease of COVID is inflammatory driven, right? In fact, all of it is. There's no more virus left. So we need a TM7 for SARS-CoV-2.
Right. Absolutely. Right. And in fact, I think that's one of the what I view TM7 is effectively the border guards that keep they're effectively the officer corps that are keeping the troops that are patrolling the border to prevent bacteria from invading the sterile host tissue. And the TM7 is actually the officer corps that's keeping the border guards in check.
Yeah, although there's a lot of experiments that need to be done to understand what's going on there. But that's a great framework to think about the next generation of experiments.
And remember that the initial baiting experiments, in many cases, I couldn't tell you how often, but they certainly say it happens with some frequency. You get what looks like an extremely destructive relationship. You get this population crash that can then rebound. What I wish I could look at the genome before that happens and after that happens to see what's changed in both sides. And I think that work can be done. And the way that these folks think at the Foresight, Professor Bohr and all of his wonderful colleagues, I would argue that they're probably thinking about that more than I am. Now, this is because this idea of evolution in a particular niche is very interesting to me. I'm teaching about it in class. We're doing a lab associated with it. So maybe my mind is too much on it. But what we are is a bunch of different ecosystems walking around. And then we have lots of passengers. And so the idea of having a difference when two different organisms are together, I think is really critical. So many times we look at one organism isolated, colonies on plates, that kind of thing. That's not how they live.
Nope.
So hats off to them for studying the three different players.
Yes.
That's great.
Yeah. Thanks, Mark.
You're very welcome.
Thank you. Fascinating as always, the work and the presentation.
Oh, yes.
All right. That is TWiM number 254. You can find the show notes at microbe.tv/TWiM. Send us your questions and comments, TWiM@microbe.tv. If you like what we do, consider supporting us, microbe.tv/contribute. Mark Martin's at the University of Puget Sound. Thank you, Mark. Good to see you.
You're very welcome.
Michele Swanson's at the University of Michigan, Ann Arbor. Thanks, Michele.
Thank you.
Michael Schmidt at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
And Elio Schaechter's at Small Things Considered. Good to see you, Elio.
Better late than never, huh?
You bet. You bet. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.
Boom.
Boom.